Hi, this is Dr. Manikandan from Department of Life Science, Christu Jayanti College, Bangalore. Today, in this lecture, I will be discussing about the southern blotting. What is blotting? Transfer of nucleic acids such as DNA and RNA and even proteins to a solid membranous support such as nitrocellulose membrane or nylon membrane is called as blotting. What is southern blotting? When fragments of DNA molecules separated by gel electrophoresis are transferred to the nylon membrane or nitrocellulose membrane, such a process is called as southern blotting. Based on this southern blotting, the other blotting techniques have emerged from this method, which were termed as northern for the RNA transfer, western for the protein transfer, and to study the post-translational protein modification, the blotting technique is called as eastern blotting, and to study the DNA protein interaction, the blotting technique is called as southern western blotting. Southern blotting is a hybridization technique and was introduced by the scientist called Edwin Southern in 1975. This method is basically used to detect the specific sequence of the DNA in that pool of DNA clones. This method reveals an information about the DNA identity, its size, and its abundance in the genome. And this southern blotting technique is based on the principle of separation of DNA fragments by the gel electrophoresis and hybridization by using gene-specific probe of the target DNA. Moving on, the following are the major steps involved in southern blotting technique. The first and foremost technique is an isolation and purification of DNA from the source sample. Second step is digestion of DNA by using restriction enzymes. Third step is separating the digested DNA fragment on to the agrogel electrophoresis. Fourth step is transfer of digested DNA fragments from the gel onto the nylon membrane or nitrocellulose. The fifth step is pre-hybridization, that is by treating the nylon membrane with a pre-hybridization solution or a blocking solution to block the non-specific interaction of the probe onto the membrane and also to reduce the background noise of the membrane during detection. The sixth step is preparation of probe. Probe is nothing but a short oligonucleotide DNA sequence which ranges from 100 to 500 nucleotides single-stranded and can be either radio-labeled using radioactive isotope or non-radio-labeled by using digoxygenin or a biotin streptavidin labeled probe. The next step is hybridization, incubating the blot with labeled probe. Last step is detection of the probe, detecting the hybridized area of the probe and the DNA sequence, target DNA sequence, by exposing the membrane to the X-ray film. Let us see the steps involved in southern blotting in detail. Step 1. DNA isolation, purification and restriction digestion. Extract the DNA from the target cell. The cell will be either lysed by using a detergent or by enzymatic treatment. The lysed sample will contain the DNA, proteins and other debris. The DNA will be purified by using PCI, trisaturated phenol, chloroform and isoamyl alcohol, and the DNA will be precipitated by using alcohol precipitation. Next process is fragmentation of the DNA by using a suitable restriction enzyme. Restriction enzyme cuts the DNA at specific DNA sites and generates the DNA fragments. Step 2. Electrophoresis of the separated DNA fragments. The desired DNA fragments will be separated on the gel electrophoresis. The DNA will be separated based on the size and the charge during electrophoresis. Step 3 is denaturation. In order to facilitate the binding of single-stranded DNA probe onto the target DNA sequence, the digested DNA fragments has to be separated into a single strand. This is achieved by using alkali NaOH or acid HCl to denature the double-stranded digested DNA fragments. Step 4 is blotting. Blotting is a transfer of fragmented and denatured DNA sequence to the nitrocellulose membrane or a nylon membrane. The separated strands of DNA is transferred to the positively charged membrane by the process of blotting. This process is carried out either by capillary blotting or electroblotting. Capillary blotting is a manual method of DNA transfer from the gel to the membrane, and this method takes 18 hours to 24 hours. Whereas electroblotting uses an electric current to pass the DNA fragment from the gel onto the membrane, and this process usually takes 2 to 3 hours. The success rate of transfer of DNA from gel onto the membrane by capillary blotting is less, whereas the electroblotting, the success rate is 100%. Step 5. Baking and blocking the membrane. After the transfer of DNA from the gel onto the membrane is complete, immobilize the DNA onto the membrane by either UV cross-linking or by baking at 80 degrees Celsius in a hybridization oven. UV cross-linking is usually preferred, that covalently binds the DNA onto the membrane and also increases the hybridization signal during reduction. Another option is by baking at 80 degrees Celsius in hybridization oven that non-covalently links the DNA onto the membrane. Once the DNA is immobilized onto the membrane, either by UV cross-linking or by baking at 80 degrees Celsius, the membrane is treated with a pre-hybridization buffer, or also called as a blocking solution, which contains casein or a bovine serum albumin, which saturates all the binding site of the membrane in order to prevent the non-specific binding of probe onto the membrane. So the step 6 is hybridization with the gene-specific labeled probe. Probe, as I already mentioned, is a short oligonucleotide DNA sequence which is single-stranded, ranges from 100 to 500 nucleotide. Usually the probes are labeled with a marker so that can be detected after hybridization. The probes are labeled by either using radioactive labeling or by using non-radioactive labeling. Radioactive labeling in using radioactive isotopes, and non-radioactive labeling is usually achieved by attaching the chemicals such as digoxygenin or by using biotin streptavidin labeling. The labeled probes contains a complementary DNA sequence to the targeted DNA of interest. So when we add the probe binds to the complementary DNA on the membrane and binds onto it by hybridization. Since the probe is very specific to the targeted DNA of interest, it specifically binds to the target DNA of interest onto the membrane. Step 7 is the detection and visualization of the hybridized area on the membrane. In the detection step, hybridization of the probe to the target DNA in a sample is identified based on what type of labeled probes that are used in the experiment. The radio-labeled probes is detected by using autoradiography. In the non-radioactive labeled probe are typically detected by incubating the membrane onto the chemiluminescent substrate and exposing the membrane or a blot to the X-ray film. For an example, if non-radioactive labeled probes such as digoxin labeled probe is used, then the antibody, which is very specific to the digoxygenin, which is conjugated with an enzyme, is used for the detection of the hybridized area. When the chemiluminescent substrate is added, the enzyme, which is conjugated with an antibody, reacts with the chemiluminescent substrate and the signal will be detected on an X-ray film. The detection time is approximately 1 to 48 hours, which depends upon the nature of the probe and the concentration of the probe. Specific band will be seen on an X-ray film, which tells us the presence of specific DNA sequence in the given DNA sample. Let us know about various applications of Southern blotting. Southern blotting technique is used to detect the presence of a particular gene in a given sample or a pool of DNA clones. It is used to analyze the genetic pattern of an individual and also used to compare the pattern between the individual, such as DNA fingerprinting, paternity testing, etc. It is used in genetic engineering, where it is used to identify the desired gene of interest in a given organism. It is used in restriction fragment length polymorphism. It is used to identify the different type of mutations or gene arrangement in DNA sequence. It is used in diagnosis of disease which is caused by genetic defects. It is used to identify the infectious agents. It is used in several other areas, including the gene discovery, mapping, evolution, developmental studies, diagnostics, and forensics. It is to determine the molecular weight of restriction fragment and is also used to measure the relative amounts in a different samples. I hope you find this lecture on southern blotting is useful. Thank you for watching, guys.